Hello, it's time to now discuss fundamentals of the nervous system and nervous tissue. This is chapter 14 in your anatomy book. The nervous system is actually referred to as the nervous organ system, in which it would be comprised of all tissue types, but it primarily is composed of nervous tissue, neurons and glial cells. In this chapter, we are going to first explore some terminology, then I'll review the organization of the nervous system. We'll review some cytology of nervous tissue. We'll discuss myelination and what a nerve is and synapses. First, some very important terms for you to get down. I suggest making flashcards of these. You should first understand what a neuron is. It is the functional unit of the nervous system. It has basic anatomy, things like soma or perikaryon, dendrites, axon hillock, axon, and the terminal bouton or synaptic terminus. It depends on your textbook what they call it. A nerve is a collection of axons, just a collection of axons outside of the central nervous system. Ganglion, or plural ganglia, is a collection of neuronal cell bodies outside of the central nervous system. If I say terminate, if I say a nerve terminates on something, I am really referring to what does it innervate onto? What will be the downstream cell that nerve is communicating to? A synapse is a functional connection between an upstream neuron and the downstream target cell. This downstream target cell could be another neuron, or as we learned in our previous chapters, it could be a skeletal muscle fiber. So the synapse is a functional communication between a neuron and the downstream cell. A stimulus. A stimulus is something that can change a cell's, particularly a neuron's, membrane potential. Those of you in my class, we have learned about different kinds of stimuli, and we discussed or used words to say the cell membrane would become less negative, more positive. Remember this? So a positive stimulus or a depolarizing stimulus is something that would get the cell closer to its threshold to spike in action potential. There is such a thing as an inhibitory stimulus, in which case it would make the cell become more negative, further away from threshold. So again, excitatory stimuli or positive stimuli and inhibitory stimuli. Integration refers to a tallying, a summation of sorts that the central nervous system, which is your brain and spinal cord, will have to utilize. You see, there's a whole bunch of incoming information, and your brain and spinal cord must make an appropriate response to that. So they basically need to gather all this information. As a parent, when my two boys come to me and they're each saying, "He did it, he did it, he did it," I feel like I'm an integration center. I'll split them both up, get their sides of the stories, then I'll try and get a third objective, and I take it all in. I'm trying to be a good judge. So that's what the brain and spinal cord is going to do. Effector cells. Effector cells are cells in the body that do something. So the basic effector cells of the body include smooth muscle, cardiac muscle, glands, skeletal muscle, and now adipose tissue. Clearly, not all of those effector cells are voluntary. So we're going to talk about which are involuntary and which are voluntary in just a moment. Motor output. When I say motor output, I want you to understand that that is an outgoing command. It's an outgoing command to target your effectors of your body. Again, organs or glands or cells even that will respond to the command of the central nervous system. Voluntary effectors include skeletal muscle. Those are the ones you have conscious control over. Involuntary effectors would include glands like your pancreas. You don't have control over that. smooth muscle, like the muscle that lines the blood vessels, cardiac muscle, and adipose tissue. I'm really sad over adipose tissue not being a voluntary effector. I think that would be amazing if we had control over that. I would definitely mobilize my adipose tissue to different regions of my body, depending on the day's events. Am I going on a date or a 6-mile run? Electrochemical communication refers to the way that neurons can send signals. They use initially chemical response. They bind to chemical signals, then they transduce that chemical signal, potentially, into an electrical one, which is then retransduced back into a chemical communication. Of course, I'm referring to a dendrite, say, binding to a neurotransmitter, this being an excitatory neurotransmitter, so the cell gets to threshold and spikes an action potential. The action potential would be the electrical form of communication. And then at the terminal bouton, this action potential is again translated into a chemical release of neurotransmitters. As we learned from our skeletal muscle unit, action potentials are all or none. That means, as we use the gun as an analogy, when you pull the trigger, there is a trigger point. And when you reach that trigger point, the bullet is fired with the same force from trigger pulling to trigger pulling. And there's no such thing as killing someone softly by, slowly pulling the trigger. If you reach the trigger mechanism, that bullet is fired with the same force. So action potentials in neurons are the same thing. Once you get that bullet fired, it goes. An action potential is marked by the opening of the sodium voltage gated channels. And once one opens, they will all open. Graded potentials, in striking contrast to action potentials, are either depolarizations or hyperpolarizations of the soma, and they dissipate with time and distance. And two famous scientists, Hodgkin and Huxley, were the ones that used a squid axon to determine between graded potentials and action potentials. So it's time to review our basic anatomical divisions. We have two main divisions, central nervous system and peripheral nervous system. The central nervous system is composed of the brain and spinal cord. That's it. If you are 1/10 of a millimeter outside of the spinal cord, then you are no longer in the CNS. You will now be in the peripheral nervous system. The peripheral nervous system consists of cranial nerves, spinal nerves, and their ganglia. So that's very important that you understand that. Cranial nerves, spinal nerves. Nerve is a collection of axons outside of the central nervous system. Ganglia, or ganglion for singular, refers to a collection of cell bodies outside the central nervous system, and that would be neuronal cell bodies found outside of the central nervous system. We can divide the nervous system not only into anatomical divisions, but as shown here, we could separate it into functional divisions. There's either sensory or incoming information, or outgoing commands. That would be your motor system. So sensory neurons bring action potentials toward the brain and spinal cord. Another way of saying this is that they're afferent. It's very hard for you to hear my pronunciation and enunciation, so I am going to say afferent, so you can distinguish between an incoming command versus efferent or efferent, which is an outgoing command. So your incoming sensory information can be somatic, and that means you have conscious awareness of it. Let's say pressure and vibration. These are sensory information that's coming from your joints, skeletal muscles, skin, special senses. You're consciously aware of this. Visceral sensory is information from viscera. Viscera you don't have voluntary control over. So a lot of this sensory information you are not necessarily consciously aware of. When I speak of motor nervous system, again this is efferent, outgoing commands, outgoing action potentials. And these nerves are going to target effectors, either voluntary, which are your skeletal muscle, that's your somatic motor nervous system, or your involuntary effectors, which would be under the control of a special nervous system called the autonomic nervous system. This would be nerves that innervate cardiac muscle, smooth muscle, glands, and not shown on this but should be included, adipose tissue. So let's review the anatomy of a neuron. Of course, we have the receiving side as shown in this picture. These are the dendrites. And remember from class, what kind of receptors do we find on dendrites? We find primarily the ligand-gated channels. I gave you one example from class for anatomy, and that was the nicotinic acetylcholine receptors. Those are ligand-gated. And to remind you, that means it's like a lock and key mechanism. And the ligand clearly would be a neurotransmitter released from some other upstream cell not shown on this picture. If it is a neurotransmitter that depolarizes the cell, then the inside of the cell would become less negative, more positive. And depending on how many neurotransmitters were released, how many ligand-gated channels bind to those neurotransmitters, and how long they were open, we can get more and more positive charge accumulating in the cell, and this could even diffuse towards the cell body, or perikaryon, and potentially reach the axon hillock. And the axon hillock, as shown in this picture, is the junction between the cell body and the axon. And its anatomical importance is that this marks the beginning where you find voltage-gated channels. And remember, those open when the cell membrane potential changes enough to open the first voltage-gated channels. And remember from class, sodium voltage-gated channels require the least amount of membrane potential change. They are the ones that open first. And we refer to this as the dance of the gates. First sodium, then the potassium voltage-gated channels. So that's what we mean by excitability. Neurons can respond to stimuli. Neurotransmitters binding to receptors. That's a response to stimuli. They're also conductive. They can spread their electrical signals, in particular down the axon to the synaptic knobs or synaptic terminals or terminal boutons. And they secrete down at the terminal boutons or synaptic knobs. We have synaptic vesicles that can fuse an exocytose neurotransmitter. So altogether, we say that neurons can have an electrochemical response to stimuli. And when we speak of an action potential, sometimes textbooks refer to this as a nerve impulse. And to remind you from class that down at the synaptic knobs, we have a third class of voltage-gated channels called the calcium voltage-gated channels. And when they open, synaptic vesicles fuse an exocytose neurotransmitter. Just to do it one more time and remind you that the neuron is the functional unit of the nervous system. It is certainly the most abundant cell type in the nervous system. Again, it has a receiving side, that would be the dendrites and somas. This is where you would find the ligand-gated channels. These are channels and receptors that bind to neurotransmitter, and when the neurotransmitter is bound, a channel opens. Depending on the kind of receptor and channel that are opened, the cell can either become less negative, more positive, that's depolarization, or it could become hyperpolarized, more negative. If it becomes depolarized, less negative, more positive enough, then at the axon hillock we see the first voltage-gated channel open. And the variety of this voltage-gated channel that opens first is sodium. As the sodium voltage-gated channels open all along the axon, the inside of the axoplasm, that's a fancy way of saying the cytoplasm in the axon, becomes extremely positive. This is a reversal, a temporary reversal of resting membrane potential. This is known as a nerve impulse or action potential. On the heels of the sodium voltage-gated channels opening are the potassium voltage-gated channels, and we learned from class that the electrochemical gradient for potassium would be out of the cell. And this would be a loss of positive charge and so the inside of the cell would go back to a negative value. When this action potential reaches the synaptic vesicles or synaptic knobs, then the calcium voltage-gated channels open, and when they open, calcium flows into the cytoplasm, and this is the molecular trigger to cause synaptic vesicles to fuse and exocytose neurotransmitter onto the next cell. There are three major structural classes of neurons. The picture from top to bottom, top picture is unipolar, middle picture is bipolar, and the last picture is multipolar. I'm going to start the discussion of multipolar neurons. Multipolar neurons are the most common of the neuronal types. They have, as you can see from this picture, many dendrites. Now, your motor neurons, your somatic motor neurons, for example, as well as your interneurons, fall into this category. Bipolar neurons are less common. We see them in particular in the retina and used by the olfactory system. You can see in this middle picture that there is a branch of dendrites that merge to form a common dendrite, which carries an impulse, a depolarization towards the cell body, and then this depolarization, if it reaches threshold, there's an action potential carried away from the cell body toward the synaptic terminus. In the top picture, I'm showing you a unipolar, also oftentimes referred to as pseudo-unipolar. This is a common neuron for sensory neurons, or a common type of neuron for sensory neurons. It really is a big axon. I want you to hold your arms out to your sides, abduct them out, and then bring your brachii together and your antebrachium. So bring your arms together, but have your arms still, your hands, facing away from the midline. And that's how these processes developed for the pseudo-unipolar. They started out as bipolar. Look at the middle picture, and then the two processes kind of came together in the middle. You see this as the short single process, but the pseudo-unipolar is very different because the two processes, the peripheral and central, are regarded as by physiologists as being a true axon. So you see voltage-gated channels here, and the dendrites are shown out towards the left side of this picture. So the peripheral process carries an action potential towards the cell body, and then the central process carries the action potential from the cell body into the brain or spinal cord. Neurons can also be classified according to their function, and I've already referred to this in an earlier slide. They can be sensory or afferent, afferent, that's the improper way to pronunciate it. The afferent, they are always bringing information to the brain. They're carrying sensory information. Does the chair hurt that you're sitting in? Does it hurt your behind? Did you just give yourself a paper cut? All of that is information sent to your brain for higher order processing. Then there are interneurons. These interneurons are always fully contained in the central nervous system. No part of them is outside the CNS. And these interneurons their functional use is that they are the integration neurons. These are the ones that have to retrieve the information. Remember, they're like me, the parent. They have to collect intel, right? Make a good decision and then send that output command. 90% of the neurons in your body are of this kind. Then there are motor neurons. Motor neurons will carry an output or a command out to effector cells, either voluntary or involuntary. So they innervate or terminate on your effectors. If it's a somatic motor neuron, then it will terminate on your voluntary effectors of skeletal muscle. If it's a visceral motor neuron, then this tells you it's going to terminate on your involuntary effectors, smooth muscle, cardiac muscle, glands, and you should add adipose tissue. clearly, if we put all three of those functional neurons together, we recreate the 5 components of a reflex arc. Number 1, shown here, you need some sort of receptor, something that can respond to some sort of external stimulus, like you stepping on a nail, as shown in this picture. So that would be a pain receptor that would detect that stimulus, as opposed to a thermoreceptor would not detect that stimulus. And then you need the receptor to be able to send or transmit an action potential to the brain or spinal cord. Well, clearly we would need a nerve to do this. So you need your stimulus to activate your receptor, and then the receptor has to be able to communicate to a nerve, and this nerve has to carry the action potential toward the brain or spinal cord. So this would be a sensory or afferent neuron. Then, as shown here for number 3, we see the blue neuron synapsing with the purple interneuron. And the interneuron is fully contained in the central nervous system. It's retrieving intel, it's getting the information, and then it has to integrate this, think about it, respond to it, and send an output command. And so we see the purple interneuron sending an output command out of the spinal cord, out of the brain as well, through this orange motor neuron. So we need a nerve to carry an action potential out of the brain or spinal cord. And then lastly, number 5, you need an effector. You need something to respond to that command. Clearly, if you've just punctured your hand or foot onto a nail, we don't keep stepping on it or pushing our hand on it to drive that nail in deeper. We would use our withdrawal reflex, right? Otherwise, that doesn't make sense. We're going to put ourselves in harm's way. No, we should get out of that danger. So here's a nice chart that shows the structural and functional classifications of neurons. Structural meaning how many dendrites do they have? How many extensions from the cell body do they have? Is it unipolar, bipolar, multipolar? Functional classification means which way is it carrying the action potential? To the central nervous system? Away from the central nervous system? Or is it the neuron that has to integrate between these two neurons, retrieve the information, and then send an appropriate output command? That would be the job of the interneuron. Alright, in our next segment, we continue on talking about more terminology regarding nerves and the nervous system, as well as support cells.